Protoplast culture in plant tissue culture. First, we know what is mean of protoplast. Below the cell wall, all parts are called protoplast. Below the plant cell wall, all parts are called protoplast. The protoplast contains plasma membrane, nucleus, cytoplasm, and all cell organelles. That protoplasts are selected from plants and cultured on the nutrient medium to develop new plantlets. That new plantlets are 100% identical to the parental plants. See, the protoplast culture mainly used in the sexually they cannot produce the next generations. Which plants are sexually they cannot produce in the next generations? That plants are will going to endangered species. That plants are cultured on the nutrient medium with the help of protoplast culture. See, protoplast culture, it is an advanced technology in plant tissue culture. See, it is one potted plant. The potted plant, healthy in conditions, it cannot produce sexually next generations. From these plants, we can easily produce more number of plantlets, more number of similar plantlets from the parental plants by the protoplast culture. See, during the protoplast culture, we selected healthy potted plants or healthy environment plant. From that plant, we selected healthy leaves. From that healthy leaves, we selected cells, isolated cells. They are transferred onto the nutrient medium. On this nutrient medium, they are produced callus. When we added to the growth hormones to the callus, they are developed into organogenesis. By the organogenesis, they can develop into root and shoots. See, these plantlets are 100% similar to the parental plants. It is the main concept of the protoplast culture. Now we discussed what are the steps in the protoplast culture. See, here it is in this protoplast culture. First one is isolation of protoplast. Second one is culturing the isolated protoplast. Now first we discussed about isolation of protoplast. How to select isolation of protoplast means how to select protoplast from the selected plantlets or selected plants. Here it is, the isolation of protoplast generally collected by two methods. One is mechanical method, second one is enzymatic method. First method is mechanical method, second one is enzymatic method. Now first we discussed protoplast isolation by mechanical method. See, in this mechanical method, cutting the cell walls is under a microscope using microscopes or sharp razors. Cutting of the cell walls is under a microscope using microsepals or sharp razors. In this process, it takes more time to this mechanical method, and also it is take more time in this mechanical method. We collect less amount of healthy protoplast only. Less amount of healthy protoplasts are collected by mechanical method. And the next one is enzymatic method. Enzymatic method is an easy method to selection of protoplast from the parental plant. See, in this enzymatic method, we used some enzymes. The enzymes are called macerozymes. The macerozymes is the composition of cellulase, hemicellulase, pectinase. These are the enzymes used in the selection of protoplast. That all enzymes are combinedly called macerozymes. See, macerozymes are used in the enzymatic method. The macerozymes are available in commercially in our local markets. They are isolated mostly from the fungi. They means the macerozymes. The macerozymes are isolated mostly from fungi. The macerozymes are commercially available in our local markets. Now, what are the steps in the enzymatic method? See, in enzymatic methods, steps. The first step is surface sterilization of leaves with 70% of ethyl alcohol. See, one important point in this protoplast culture, that protoplasts are selected from only leaves. That leaves are sterilized. The leaf surface are sterilized with 70% of ethyl alcohol. 70% of ethyl alcohol solution. Later, washing of leaves with the distilled waters. Washing of leaves with the distilled water. Later, peeling of leaf pieces only from lower epidermis. Peeling of leaf pieces only from lower epidermis. See, lower epidermis pieces are collected from the leaves. Later, placing leaf pieces, lower epidermal leaf pieces are placing in a 13% of mannitol solution for plasmolysis. The lower epidermal leaf pieces are kept in a 13% of mannitol solution for plasmolysis. This mannitol solution, including lower epidermal peels, kept on shaker water bath, shaken in water bath machine for the plasmolysis. After placing leaf peels in cell wall degrading MJ mixture. See, after plasmolysis, the cell wall degrade MJ mixture is added to the leaf peels. Placing of leaf peels in cell wall degrading MJ mixture That MJ mixture, cell wall degrading MJ mixture contains pectinase, cellulase, hemicellulase. These are also called mass serozymes enzymes. Mass serozymes. Pectinase, cellulase, hemicellulase. These enzymes are called mass serozymes. The mass serozymes are called cell wall degrading enzymes. See, the leaf peels are placing in mass serozyme chemical and incubated 24 hours at 25 degrees Celsius temperature. After this time, the cell wall is degraded. The cell walls are degraded. Only we identified protoplast in the mixture. After filtering the above material by the centrifuged method, after centrifuged method, filtering the above material. Above material means cell walls are degraded. They are contains high molecular weight. They are present at the lower part of centrifuged test tube. And above these cell wall degrading mixture, clean protoplast are identified. That protoplast and cell wall degrading mixture is filtered and separation and wash with sorbitol. The selected protoplast by the filtering called separation. The separation of protoplasts are washing with sorbitol solution and placing in 20% of sucrose. Placing in 20% of sucrose. See, by the filtering mechanism, by filtering process, we selected or separated protoplasts. That protoplasts are washed with sorbitol solution and kept in or placing in 20% of sucrose solution. Later, pipette out selected protoplasts after centrifuged method and placing in cultured medium. It is the selection of protoplast procedure by enzymatic method. Now, second step is culturing the protoplast, culturing the isolated protoplast, on the nutrient medium. It is the second step or second procedure, second step of the protoplast culture. The protoplast can be cultured in agar embedding medium. Protoplast cultured in agar embedding medium. The protoplast and agar mixture with equal volume take in a petri dish. Selection of protoplast and agar mixture with equal volume, they are taken in a petri dish. The above mixture, what is the above mixture? It is a selection of protoplast and agar embedding medium. The agar embedding nutrient medium taken with equal volume, they are taken in the petri dish. The petri dish is inverted after the medium is solidified. See children, before the solidification of nutrient medium, the petri dish is in normal condition, normal position towards upright direction. In that petri dish, we take agar embedded mixture with equal volume of selected protoplast. After few minutes, that nutrient medium, agar embedding nutrient medium is modified into solidified. After solidified, that petri dish is convert into change into inverted direction. Change into inverted direction. The protoplasts are synthesized cell walls. After petri dish inverted, the protoplasts are synthesized new cell walls around the protoplast. Then they are incubated at 25 degrees Celsius temperature. After development of callus, we add growth hormones. Development of callus, we add growth hormones. They are developed into roots and shoots. This process is called organogenesis. Roots and shoots are developed after adding of growth hormones. This step is called organogenesis. After organogenesis, we can easily identify some plantlets on the petri dish. That plantlets are transferred to the plantation on the soil, or before plantation or outside, they are planted on the pots. The pots are kept in proper environment. In that proper environment, the plantlets are comfortable. Later, that plantlets are plantation in the normal soil at outer environment. That plantlets are 100% identical to the selected parental plants. That plantlets are 100% identical to the selected parental plants. It is the protoplast culture. It is mainly used in the which plants are sexually cannot produce next generations. That plants are cultured on the tissue culture procedure with the help of protoplast culture. If the protoplast culture is absent, cannot applicable to these plants, automatically these plants are reached to the endangered species, then after extincted. So it is the main important for this type of plants. It is the process and main use of the protoplast culture.